This method can help to answer key questions in the field of gene therapy, oncology, and genetic vaccination. It facilitates understanding and modulating of vector-host interactions of adenovirus gene transfer vectors, the most frequently used vector type in clinical trials up to date.
The main advantage of this technique is that adenovirus capsids can be specifically modified in a highly defined manner at selected positions and to variable extent by using simple chemistry.
The implications of this technique extends towards therapy because vector-host modification can not only be analyzed but also modulated in a patient-oriented way.
Though this method provides insight into specific vector-host interactions, it may also be used for labeling and tracking of virus particles in living organisms, such as mice.
Generally, individuals new to this method will struggle with the unique combination of virology and organic chemistry that requires practical knowledge from both fields.
Visual demonstration of this technique is critical because here methods from organic chemistry meet methods from cell biology and virology, including field-specific handling tricks.
To begin this procedure, prepare all buffers as outlined in the text protocol. Weigh 1 milliliter of each gradient buffer to check its density. After sterilizing all of the buffers, sparge the buffers containing TCEP with argon gas for about 30 seconds to prevent oxidation of the TCEP. Store the vials containing the solid powdered coupling moiety in larger tubes.